Wanner. I'm professor at UCLA Neuroscience. And I want to first thank the organizer for having me on in this really interesting meeting. Today, I will be talking about tracking and treating astrocyte injury and recovery after neurotrauma. We have a patent on astrocyte traumatomes and neurotrauma biomarkers. I will share with you basic facts on neurotraumas and our target, the astrocytes. We are using a human in vitro trauma model, and we have devised a novel panel of injury-defined biomarkers that I'll introduce, and then share with you findings using a drug that can improve astrocyte survival. Traumatic brain injury is an impact to the brain due to whiplash or a force that can cause deformation and shear forces, and in fact, injure brain cells that have biochemical changes. The impact can have secondary sequelae and cause neurodegeneration and chronic cognitive and psychological deficits. Neurotrauma has a huge global burden. New estimates show that 64 to 74 million people suffer a traumatic brain injury per year worldwide. And there's an additional 500,000 people suffering a devastating spinal cord injury per year worldwide. These are new estimates that go beyond hospitalization numbers. We are studying astrocytes because they are pivotal for health and disease and injury. Astrocytes constitute a big part of the blood-brain barrier, and they're also important for the function of the neurovascular unit. As we will hear from Pierre Magistretti shortly, they provide metabolites and neurotransmitters to the neurons, and they have very fine endings wrapping up around blood vessels, as you see here on the left. Now, after injury, you can imagine how these essential functions are jeopardized. We see metabolic crisis, loss of ion homeostasis, and the cytotoxic edema that is actually associated with astrocyte death. On the flip side, we also see that astrocytes respond, become reactive, associate with inflammatory cells, and form the glial scar that you see here on the right in my studies with Mike Sofroniev. The glial scar is actually neuroprotective. We are studying human neocortical astrocytes from donated specimen, and you see here differentiated human astrocytes. And this is the picture just after pressure pull stretching, where we basically mimic the deformation because the cells are on rubber membranes. You see cell amputations, you see vesicles and blabbing, but you also see a transformation of the astrocytes, now building numerous novel processes which astrocytes are so famous for. The human astrocytes can have up to 50 or more processes in these cultures, and that's a hallmark of human astrocytes. We see here elongated processes, and here was the cell I just showed you, but in a similar area right next to it, we see a similar event of a transformation of an astrocyte, here forming a very long process. And this basically demonstrates that there is reactive responses to mechanical trauma by stretching. Of course, the same culture also showed all the wounding, and this is identified by using propydium iodine that actually labels all the wounded astrocytes in this culture after stretching. And the kinetic of this stretching using this dye shows that here we see the dye uptake in positive cells. These are the wounded early cells. They still have intact nuclei. And then by 2 days, there's increased cell death, which is shown by the full pycnosis of these cells. Now, this culture model shows cell wounding, energy deficit, and also cell death, as well as this transformation into the reactive state. Now, what are the release profiles? We're interested in finding out whether we can identify different stages by different proteins in the fluid. we did initial 2D gels, and what you see in red are all the proteins that are significantly released very early on. This was a surprise when we looked at what these proteins were. They were actually metabolic proteins. And when comparing with the cellular proteins, these are shown here with the turquoise bars, you see that the metabolic proteins are overrepresented. So there is a dominance of spilling metabolic that leaves the cells depleted of metabolic activities. We repeated these studies using isobaric mass tag, a different mass spectrometry approach, again quantifying protein release, and in human astrocytes, metabolic profiles are also prevalent, and you see specifically every enzyme of the glycolytic pathway is enriched in astrocytes and is released significantly. Here, one example is the aldolase C, stained here in green. You see this blabbing of the wounded cells, and these enzymes are basically gone out of the cell, leaving the cell deprived. This is actually a hallmark of metabolic crisis, impaired glucose use, and ATP deficit. So this really matters. The early cytosolic proteins, but when looking at by 24 hours in these more vulnerable mouse astrocytes, we found cytoskeleton and cytoskeleton-associated proteins, and these are the profile of cell death. Now, the reactive astrocytes, and we found that in the human astrocytes by the 2-day profile, show mainly extracellular matrix proteins, vesicle proteins, and later inflammatory proteins. So we have different profiles, and we were wondering, can we use any of this to devise novel biomarkers? And to do this, we, of course, overlapped our in vitro traumatome with traumatome from TBI patients, and then selected for astrocyte enrichment and brain-specific proteins. This is how we got to the panel that shows aldolase C as one of the most brain-enriched proteins, glutamine synthetase, brain lipid-binding protein, the PEA15, and smaller fragments of GFAP. Now, these proteins do show the differences between cell wounding and cell death. Aldol-C's early released GFAP fragments that you see here on the Western blot here on the left are generated only later. And in fact, they do associate with the percent of cell death in the culture significantly. And the Aldol-C levels do significantly associate with the percent of cell wounding earlier in these cultures. And we are capitalizing on these proteins, and we're making ELISAs to arrive at the blood test for concussion and for ruling out more severe damage. So I've showed you these profiles of cell wounding and cell death in astrocytes after stretching. And in the next part, we really want to hone in on those energy-deprived astrocytes and see if there's a therapeutic way of improving their survival. We have done this in our Dana Foundation Award, where we tested the efficacy of a novel drug. We partnered up with Astrocyte Pharmaceuticals using an adenosine agonist, which is AST004. It's a purinergic receptor agonist. It penetrates the blood-brain barrier, and the work from Jim Lechleiter has really shown that it improves ion homeostasis, that it can basically reduce oxidative stress and improves overall outcome. When we use this in our culture model, we basically made sure to use different doses. We made sure to use unbiased imaging approach, automated analysis. And what we're finding is that this massive cell death early on is actually attenuated with various doses of the drug, almost back to baseline level. We do see differences. We find that actually the earlier stages of cell death, where the pycnosis is not yet fully executed, is also attenuated. And this really suggests that the drug can basically help reseal the cell and stop the progression to cell death. We also found that the GFAP staining in these very wounded cells is really pathological. We see basically that the cytoskeletal structure goes away and we see non-fibrous GFAP, and that is in almost 100% associated with the leaky cells that are undergoing cell death. The drug AST004 was able to significantly reduce also the profiles of this pathological GFAP. In addition, though, we do find that the drug can also attenuate the fragments of GFAP in the culture fluid. So GFAP breakdown and disassembly is improved when the drug is acting on the cells, helping them survive. This is again a picture that shows astrocytes in their healthy state with GFAP cytoskeletal, and on the right, the damage after stretching. And in this next picture here, I show you the improved situation in the presence of the A3 agonist. And on the right, we partially reverse the effect of the drug by adding the MRS 1523 antagonist of A3 receptors. This all next to one another, I think really should show you very well how AST is able to improve the overall cell death rate. Let me just show this to you. Here you see the reduction of the PI uptake and cell death that is partially reversed by using this MRS antagonist. The antagonist also reversed the fragmentation of GFAP, and what you end up with is astrocytes with more intact GFAP. Now, these are really preliminary findings, but we are basically having a working model of the neuroprotective action of AST004. AST004 binds to the adenosine receptor, IP3 levels are elevated, and in the ER, calcium can be released that is rapidly sequestered into mitochondria because mitochondria and ER are very tightly next to one another. So calcium elevation in mitochondria can actually stimulate the TCI cycles because of the enzymes that are calcium dependent. And this elevates overall ATP levels, and that basically can help astrocytes to take up glutamate, to attenuate and restore ion homeostasis, and to really combat oxidative stress and excitotoxicity. So with this working model, I want to just summarize our findings. We have different phenotypes identified of released proteins after astrocyte trauma, and from that we derived our novel biomarker panel that can track astrocyte wounding and astrocyte death. The drug, the adenosine agonist AST, substantially improved cell survival and reduced the GFAP pathology. The drug was partially reversed by an antagonist, so it's acting through the A3 receptor, and we propose that we can use GFAP fragments as pharmacodynamic biomarkers to really help clinical trials in the future for traumatic brain injury patients. So with this, I want to thank my collaborators and the people in my lab, and I want to thank my collaborators from the industry, Astrocyte Pharmaceuticals, NCORE, and BrainBox, and my colleagues in the Brain Injury Research Center and the Molecular Instrumentation Center. And I'll be happy to take questions. Thank you.